Good afternoon, students. Today we discuss microbiology method and instrumentation. At the end of this module or topic, students will be able to describe the methods of measuring total sum number of microorganisms, describe the methods of counting viable cells of microorganisms, describe the methods of measuring cell mass of microorganisms, states the advantage and disadvantages of the different methods of measuring microbial growth, explain how different environmental factors affect microbial growth. So what are the measurements of microbial growth? Population growth is measured by following changes in the number of cells or changes in the level of some cellular component. This could be done by measuring the total cell number or by measuring the cell mass. So when you talk about measuring the cell mass, the total number of microbial cells can be achieved by using direct count method. In direct count method, microorganisms can be enumerated by directly counting them. The special counting chambers, such as the hematocytometer, and you see in the lab, you have it in the lab, you use it to count the microorganisms. Then these chambers are ruled from square of a known area and constructed in such a way. So we look at the diagram and see how the counting chamber looks like. We have advantages of this counting chamber. They are used in counting chamber is very inexpensive, easy to count. The information about the size and morphology of microorganisms. The disadvantage is that the microbial population may be fairly large for accuracy, so they may be jammed together. The prokaryotes are more easily counted than the eukaryotes. Now we talk about another method of counting. Counting is the membrane filter. The membrane filter, you can talk of your visualize your sieve for camo. These are what we look that looks at it's 0.2 micrometer in pores. These are when you pour the liquid, especially in liquid, when you pour the liquid, it will help to count and to filter them. The variable counting method, this is the method you use to count the variable cells. The variable cells in the sense that the cells that are active. Advantages of variable count method or plating out. These are simple and sensitive, widely used for variable count of bacteria, and you use it in food, water, and soil sample. The disadvantage is that it is selective, and the nature of growth medium and incubation condition determines which micro or which bacteria can grow and counted. Because you are bent on counting the variable count. Then advantages of variable count method of plating out is what we have stated. They are easily counted and the rest of them. The most probable number we heard of MPN techniques. So these are counting the most probable number is a statistical method based on probability theory. So you use it in this method. Multiple serial dilutions are performed to reach a point of extinction. So you will now do this, the multiple serial dilution in the lab. This is what you use as the most probable count. And in most of those that will work on food in their research with this. So look at the diagram of the direct microscopic counting chamber. And when the school reopens, if you go to the lab, tell Mr. Emmanuel to show you this particular counting chamber. We have it in the lab. Then when you talk about the measurement of cell mass, the determination of microbial dry weight can be measured, and it can be measured by the cells growing in the liquid medium are counted by centrifugation. When you centrifuge, you wash and dry, and then this is used in techniques for measuring growth of filamentous fungi. You use it a lot. Then spectrophotometry. These are mainly instruments used by those in biochemistry. This method depends on the fact that microbial cells scatter light that strike them because microbial cells in the population of roughly constant size. So the amount of scattering is directly proportional to the biomass of the cell. So you use the amount of the light scattered. When we have spectrophotometer in biochemistry lab, you also visualize it when you come back to physical class in the school. Then the essence of light scattering is what we measure in spectrophotometer. So factors influencing microbial growth. When you talk about the factor, the rate of microbial growth and death are greatly influenced by environmental factors or parameters. Some environmental conditions of favor rapid microbial growth, while others do not permit bacterial reproduction. Understanding the influence of environmental factors on microorganisms help in the control of microbial growth and the study of ecological distribution of microorganisms. So these factors, the factors you see in the environment, we talk about the environmental factors, the solutes and water activity, pH, oxygen level, pressure, and radiation. So these are environmental factors. Then the growth and activities of microorganisms are greatly affected by physical and chemical state of the environment. So the four key factors are temperature, pH, water, and availability of water availability. So these are what you consider when you talk about the environmental factors. So let's look at this is torpedometer estimation of bacteria. This is what you see. This is a spectrophotometer. When you come back, you look at it in the chemistry lab. We talk about the temperature. As I said, temperature is one of the key environmental factors. Temperature is the most important factor affecting the growth and survival of microorganisms. So either at a too cold or too hot environment and temperature, most microorganisms will not grow or may even die in such environment. So every mostly more there are microorganisms we see around us grow under a minimum or environment that is the middle at the optimal temperature. So every microorganism there is a minimum temperature below which growth is not possible. An optimum temperature at which growth is most rapid and maximum temperature which is not possible. So these are three temperatures. These three temperatures are called the cardinal temperature that are characteristics of a given microorganism. So let's look at the temperature classes of microorganisms. We have what we call the psychophytes, the psychophytes. These are these are organisms that grow well at 0 degrees and have an optimum growth level at 15 degrees centigrade or lower. And the maximum is around 10 degrees centigrade. They are readily isolated from Arctic and Antarctic habitat. Most sacrifices are found in the ocean, so it is one what is common in an environment. We have the cyclotrophs or facultative sacrifice. So these are the organisms that have the optimum at such level. Then we talk about the mesophytes, which I mentioned before. So most microorganisms we see around us belong to this group. These are microorganisms with growth optimum around 20 degrees to 45 degrees centigrade. So they often have temperature minimum of 15 to 20. So these are what they operate. The room temperature is around 20 to 25. So these are the organisms we see around us. And the body temperature is fairly at 37 degrees centigrade. So this the mesophyte. Most organisms belong to this. After mesophyte, we have the thermophytes. These are organisms that can grow at a higher temperature, around 55 degrees or higher. So we see some of the organisms surviving. The majority of thermophytes are prokaryotes, although a few photosynthetic protists and fungi are thermophytes. So you see more in the hot spring water and the rest of them. Then we have the extreme thermophiles or hypothermophiles. So these are prokaryotes that have growth optimum between 80 degrees to 11 degrees centigrade. So it's just within, not in a very hot sea. So these are. So you look at this diagram, you see these are the psychophytes, the psychotrophs, the mesophytes, the thermophytes, and the hyperthermophytes. You look at the temperature level. Then oxygen concentration. You know, we are dealing with the environmental factors that help in bacterial growth. The oxygen concentration. Most important, the importance of oxygen to the growth of organisms correlates with its metabolism. So based on the ability to grow in the presence of or absence of oxygen, microorganisms are classified as aerobic organisms, that is the aerobes. These are organisms that are able to grow in the presence of atmospheric oxygen. We have the anaerobes. They grow in the absence of atmospheric oxygen. Then we now have the facultative. These are organisms that do not require oxygen for growth, but grow better in its presence. They do not require, that is why they are called facultative. So they now have the aerotolerant anaerobes. These are not dependent on oxygen. They don't depend on, but they grow equally whether oxygen is present or absent. Example is Enterococcus faecalis. Then we have strict obligate anaerobes. So they do not tolerate oxygen at all, and due to its presence. So example is Clostridium pastorianum. Please see what you see mostly in food. Then we have the micro aerophiles, that is, they need a little of oxygen. These oxygen are damaged by normal atmospheric level of oxygen. So you now see, we see some example in Campylobacter. Microbial growth that may show more than one type of relationship to oxygen. A type is found among the prokaryotes and the protozoa. Fungi are normally aerobes, aerobic organisms, but a few species, particularly among the yeast, are facultative anaerobes. Facultative means they can adapt to non presence of oxygen or absence in presence of oxygen. But when you talk about obligate, means they cannot survive under that. So when you look at this diagram, you see the effects of oxygen on the growth of various types of oxygen. When you study the diagram, you understand what I have explained already. We now see the pH, the effect of pH or acidity. pH is the measure of the hydrogen ion activity of a solution, and it is defined as the negative logarithm of hydrogen concentration. We did this in our chemistry in secondary school. So in pH, the pH scale have between 0 to 14. The acidophiles, they have their growth optimum between 0 to 5.5. The neutrophiles, they grow between 5.5 to 0.8. Then we have the alkalophiles, that is the alkaline, the 0.8.0 to 11.5. The extreme alkalophiles have growth optimum at 10 or higher. In general, different microbes group, different microbes or microorganisms have characteristics. Preferably, they grow in neutrophile, that is neutral pH. Most fungi are pseudophytes between 4 to 6, and photosynthetic proteins also favor slight acidity. Many archaeas are acidophytes. They will have the solute and the water activity. This ability of microorganisms to grow over a wide range of water activity or osmotic concentration. We have the selective permeability, which we studied the osmosis, we studied the plasmosis and the rest of them. This is actually what we are talking about. The diagram shows you the cell, the organisms that grow in a solute in a higher concentration when there is salt, and where there is no, so what happens to the oxygen? It's the same theory that we have in our study of osmosis. We have radiation, which is the sun. The sun is the major source of radiation on the earth. It includes the visible sun, the ultraviolet sun, the infrared, and the radio waves. But studies have it that sunlight is the major when organisms are exposed to intensive sunlight. It mostly kills most organisms. So visible light is most conspicuous and important aspect of our environment. So organism dies easily when exposed to radiation. And it's also when you expose organism in ionizing radiation, For example, most of the organisms, like the via, the drugs that are produced are exposed to ionizing radiation in order to kill microorganisms. Remember in our previous studies when we are studying instrumentation, what we do for to kill microorganisms, sterilization and sterilization and disinfection. These are the purpose of using ionizing radiation. Now, so at the end of this study, we are able to establish the growth of micro. What are the measures through which we check the growth of the microorganism? What are the environmental factors? And how do you take care of the environmental? Or what are the needs of the? Remember, the topic is microbiology methods and instrumentation. What are their methods, the method, what they do. Remember, we go back to our learning outcome. We are now will be able to describe the method of measuring total sum of total some number of the microorganisms, and be able to describe how you use the counting chamber, the counting variable cells, in counting the cell mass, then the advantages and disadvantages of each method, and be able to explain how different environmental factors affect microorganisms. Thank you, students.